Good evening, everyone. Very good evening, everybody. Please confirm if my screen is visible and am I audible to everybody? Good evening, Rahul. Hello, Tarun. Good evening, Aishwarya. Good evening, Deepshika. So I hope you all are doing great. So we'll start the session in a minute. Let's others also join. Hello, Aditya. Very good evening. Okay, thank you for confirming, Deepshika. Good evening, Pankaj. So I hope everyone has joined now. Yeah, very good evening, Ashu. Good evening, Sahil. Glad to see you all here. So welcome to another session of Brahma series. And as you know that in this series, we discuss the CSI net questions. We discuss the questions majorly from part C, the part C questions which are asked in CSI net. And today, I have come up with the questions on topics like tetrad analysis and physical mapping. So basically, these are the topics that you study in subunit 8D of that is subunit D of unit 8, that is inheritance biology. And unit D is all about the gene mapping methods. So in the previous session, I discussed the questions about, I discussed the questions on linkage maps, and like we discussed the questions on gene order, the distance between the genes. And today we will be discussing the questions on tetrad analysis and physical mapping. So I've already covered these topics in detail in the regular classes. So I'm sure if you have attended, you know the concepts. And today you can utilize those knowledge of your concepts in solving the questions today. Hello, Sahil. Yeah, sure. I will be when I will be like when I'll be discussing the questions. Definitely, I will be telling you about the concepts behind those questions, and definitely those concepts will be covering the basics, right? So definitely, I'm sure this session will be helpful to you. So let's get started. So let's start with the very first question. And here you can see that this particular question, if you have gone through previous year question paper, so this question is a very easy question. And to begin with, I have given you a easy question from these particular topic. So this particular question is from Tetrad analysis, and it's a question of CSI net exam, and it's a part B question, right? So let me see how many of you can recall what I told you during the class. And still, if you have not attended the class, no worries. I'll tell you the concepts here. So go through the question, and I really appreciate if you all will answer the questions. See, at the end in the exam, you only have to solve the questions, right? I'm just here to help you all. The Biotechnica has come up with this Brahma series to help you all because we get a lot of questions from the students that you need the sessions on question and answer discussion, right? The questions we give in weekly test, right? Based on regular classes we conduct, right? So here, this is the platform where you can, test your knowledge and even practice questions. And even if you have any doubt, you can ask me live during the class, right? So I really appreciate you all if you answer the questions, if you take initiative and answer the questions. And even if your answer is wrong, no worries, because as I always say, it's a bit like unless and until you don't solve, how will you understand whether whatever concept you're thinking is right or wrong, you're thinking in right direction or not, right? So just answer it, okay? Don't worry about whether your answer is right or wrong. For that, I'm here to help you. So I can see some answers are already coming. Rahul, Ashu, Pramod, Suresh. So thank you. You have already answered option 3. Aishwarya, really great to see your answer. So it's answer 3. So quickly, you need not to take too much time. So the question is about segregation of alleles can occur either at anaphase 1 or anaphase 2 of meiosis. So which one of the following is an ideal model system for identifying the stage at which allelic segregation occurs? So basically, to answer this question, like it's a very direct question based, memory based question. So if you have studied about tetrad analysis, you can easily answer the question. Yeah, Anjali, very good evening. Like this is the first question I'm discussing. So you are not late. So quickly answer the question what it is. Yes, everybody is not answering the question. I can see many are here attending the sessions, but all are not answering. Why is it so? I'm sure if you are here, that means you are here with a purpose, right? So just utilize this platform and answer the questions. So most of you have answered 3 and you are absolutely right. So basically, when we talk about the tetrad analysis, I'm sure you know that the Neurospora is a very popular model organism that we use for tetrad analysis to study tetrad, right? So what is like Neurospora is a genus of ascomycete fungi, and it's a fungus where spores are arranged properly in spore sac till the 8 spore stage. So basically here when we talk about the sexual reproduction of fungi, so haploid spores, so basically here, let's say these are two different cell mating types, right? So these cells, they fuse together to form a diploid zygote. And now this diploid zygote will undergo several cell divisions like meiosis 1. So meiosis 1, as you know, it's a reduction division. So followed by meiosis 2. And that results into formation of 4 ascospores in an ascus. So what is an ascus? It's a cylindrical shaped structure which contains the spores of ascomycete fungi. So, like when you talk about the sexual reproduction of fungi, the haploid spores, they fuse to form a diploid zygote, and finally the zygote will process through meiosis and will produce a group of 4 haploid spores, which we call it as a tetrad. And these ascospores, okay, spores of ascomycete fungi is ascospores, and they are arranged in an ascus, which is a cylindrical bag-like or shape-like structure. Now, further, if the mitosis division occurs, then it results into formation of an octad. So basically, now in ascospores, there will be total 8. 8 ascospores will be there. So either you can call this structure as double tetrad, or you can call this structure as an octad, right? So here the arrangement is where 4 will be of one type ascospores and 4 will be of one type. So again, here we will consider a few cases, like we are talking about segregation of allele. So how can you study about segregation of allele? So this is a basic introduction about the life cycle of Neurospora, that two different mating types, their cells fuse to form a diploid zygote, followed by divisions, and finally you'll get an octad. And majorly the octad, which is formed by Neurospora, or tetrad formed by Neurospora, it comprises of it is orderly arranged. So we call it as ordered tetrad. Like you can see, there is no much space in the ascus, so there is no shuffling of ascospore can happen. And as a result, we say that Neurospora forms the ordered tetrad. So tetrad means four ascospores are here in an ascus, right? Now, if we talk about the segregation, so always remember that generation of ordered tetrad within an ascus in Neurospora can be studied by considering two cases. One case is when there is no crossing over, and another case is when there is crossing over. So here you can see, I told you that there is a fusion of cells of two different mating types. So we are considering one cell to be with this genotype and another cell with this genotype. So when they fuse together, they form a diploid zygote. So heterozygous it is, right? Now you can see that when this zygote undergo divisions, right? So here you can see this is the first meiotic division. So we consider that when there is no crossing over, we consider that the segregation of alleles happen during meiosis 1. This is what scientists have studied by taking Neurospora as a model organism, right? And finally, you can see that meiosis 1 followed by meiosis 2, which is like mitosis only. And then further, if meiosis 2 is followed by mitosis, so this will form these two, this will form these two, this will form these two, and this will form these two. So if we talk about the ratio you can see these four are of one type and these four are of one type. So ratio is 4 is to 4 in an octad, 2 is to 2 in a tetrad, right? And if the arrangement of the allele is like you have this type of allele over here and another allele over here, then you can get this kind of arrangement where 4 are of one type and these 4 are of another type. So it's just different in terms of arrangement. There is no difference in ratio. Ratio is 4 is to 4 only. However, when there is a crossing over, now, when there is a crossing over, we are considering only one allele here, right? Allele A. So obviously, with respect to what crossing over is happening, so we take centromere as a reference point. So we say that when crossing over is happening, it happened between locus A and the centromere. And specifically, when there is a crossover, we say that the segregation of alleles happens during meiosis 2. That is M2 segregation pattern. So here you can see this is the crossing over happening between second and third strand. So you can see this strand is non-recombinant, right? It has not participated in crossing over. But if you see the second strand and third strand, you can see these are recombinants, right? And finally, after meiosis second and mitosis, you can see that we'll get the ratio 2 is to 2 is to 2 is to 2. Like these two are of one type, these two are of one type, these two are of one type, and these two are of one type. Like if you see here, this one and this one is not same. This is just like parental, but this one is recombinant. Similarly, this one is recombinant and this is just exactly like parental, right? So ratio here is 2 is to 2 is to 2 is to 2. Now you may ask that, ma'am, the crossing over can happen between strand 1 and 3 or 1 and 4 or 2 or 4, right? So depending upon which strand is participating in the crossing over, accordingly, either we'll get 2 is to 2 is to 2 is to 2 ratio, or we can even get 2 is to 4 is to 2 ratio. So therefore, as it is mentioned or it is asked in the question that which organism is used to study the segregation of alleles, right? So it's Neurospora, and here I've already told you that studying the Neurospora or like when we are using Neurospora as a model organism, we can study that how the segregation of alleles happens in meiosis 1 or meiosis 2. Like when does the segregation of alleles happens? So answer here is option 3. Yeah, very good evening, Sunshine. You are absolutely right. It is option 3. Now moving over to the difficult question. So initially I give you the easy question. So just to increase your confidence, and further your confidence will increase in solving questions if you get the part C questions right. And it all comes with a practice. So today let's discuss some like as in every session I discuss the tough questions and the part C questions. So here is one such type of question. So please go through it and you can answer in the chat. So the following table summarizes the cross between two strains of Neurospora having alleles. So these two are the alleles. So in the previous example, I took A, right? So here it is D mentioned. Now the table shows different pattern of octad arrangement, right? That is 8 haploid spores. And the number of the ascus observed of each type, right? So here you can see the number of ascus observed mentioned in the last row. Now, based on the above, fill in the blanks from the options given below. So here you have to fill this A, B, and C. So the entire statement is that the first two columns, so these are the first two columns. First two columns are from meiosis with no crossovers between locus D and you have to fill it. And the second is that the patterns for these two columns represents dash segregation pattern. And third is that the distance between locus D and the centromere is dash map units. So just see, in one question, they have asked you everything. They have asked you about the recombination frequency, right? Like based on that, you can calculate the distance. So you have to recall the formula. Your concept regarding when do you get when the meiosis 1 or meiosis 2 segregation happens, that you have to recall the concept here. So think about it and answer the question. Okay, Rahul, you are not saying the last column is not visible. Please check the settings once, Rahul. Please, you can change your settings if it is not up to the highest pixel. Okay, I'll write it down if it is not visible for you all. Okay. So it's 115, it's 125, it's 14, it's 16, it's 17, and it's 13. I hope it's fine now. Just change your video settings also. Sometimes it happens you are not able to see the screen properly. So just change your video settings also. See, here I would like to tell you one strategy of solving such questions. First of all, always look for which, like the question is based on which organism. Is it yeast, Saccharomyces cerevisiae, or is it Neurospora? These are the two very common model organisms that, or popular model organisms that we use. Always remember, when it is about Neurospora, it's an example of order tetrad. So order tetrad cases and formulas you have to recall here. Because those who have studied tetrad analysis in my class, they know that there is another case of unordered tetrad, which is seen in yeast, right? So if the example is of yeast, then there you have to recall the formulas, the cases that we have studied in unordered tetrad analysis. So one thing is very much clear, that when you talk about Neurospora, it is about the ordered tetrad analysis, right? And students, I have just checked with I have just checked with my technical team. It is absolutely fine. I think it's at your end. So just as I have said, please check your video settings. Okay, just check your video settings once. Okay, now here I'm getting option A as an answer, option D also. So obviously all three cannot be correct, right? Any one will be correct. But thank you for answering. I really appreciate your response. Keep it up. At least you all are trying. Very good. Okay, Anjali, option 1 you are saying. Fine. So just like few minutes before, I just showed you one diagram, right? I told you that for Neurospora we consider two cases. One case when there is no crossing over, and another case when there is a crossing over, right? So just recall that point. So as I told you that if there is no crossing over, what ratio we expect? We expect 4:4 in an octal, right? However, if there is a crossover, then what ratio we expect? 2:2:2:2 or 2:4:2. So one thing is clear, that whatever table is given to us, by seeing the arrangement of the alleles, we can make out whether the we are getting which ratio, and accordingly we can say it's a case of crossover or it's a case of no crossover. And once you find out that if it's a case of crossover or no crossover, accordingly you can answer this also. You know that when there is no crossover, always meiosis 1 segregation pattern is observed. However, when there is a crossover, crossing over always happens between the locus and centromere. So here the locus that we are studying is D, right? And always the segregation pattern observed is meiosis 2, right? M2 is meiosis 2, M1 is meiosis 1. And if you are like here in the third part of that question, the C1, which we have to find out, the C blank, for that we have to calculate the recombination frequency, right? We have to calculate the distance, right? Distance of the allele. So we have to always use the formula, and the recombination frequency formula is always number of recombinants by total progeny into 100. So if you remember, in the crossover only we'll get the recombinants, right? So you can see that these two are non-recombinants, these two are non-recombinants, but these are recombinants, right? These are recombinants. So actually here, if you see, right? So these are recombinants. So some are non-recombinant, some are recombinant. So always remember the formula is half into M2 SK by total SK into 100, right? So total SK means including both non-recombinants and recombinants, and M2 SK, that is the, again, M2 SK. So see, total SK will be, you have to consider both the cases, when there is no crossing over and when there is crossing over. So if there is no crossing over, you are again getting non-recombinant. So you have to include here them as well. In M2 SK, only when there is a crossing over happening, that case you have to consider. And even in M2 SK, we are multiplying it with half because only half of the spores are recombinant. Okay. So I hope you can answer the question now. Okay, Murchana, you are saying option B. Second division if crossing occurs. Yes, so M2 segregation if crossing occurs, right? So in the question we have, let's answer it step by step. So it is first two columns are from meiosis with no crossover between locus D. And what is A here? So first two columns, just see the ratio. What is the ratio? What is the ratio you get when there is no crossing over? We get 4:4. So let's check. So yes, you can see these four are of one type, these four are of one type, right? Similarly, here you can see these are of four are of one type and these four are of one type, right? So we have, like, this is the kind of ratio, right? 4:4. And when there is no crossing over, what kind of segregation occurs? Meiosis 1, M1 type, right? And also you have to find out that when there is no crossover, so R from meiosis with no crossover between locus D and, so always we consider the crossing over with respect to centromere, right? So that means either B could be right or D could be right. Answer cannot be A, answer cannot be C, right? Then second point is that the patterns for these two columns represents what kind of segregation pattern? I already told you. It's first division segregation pattern, meiosis 1, right? So this confirms your answer. Answer is option B. Easy, right? Yes, Sunshine, Rahul, absolutely right. 4:4. No, Sunshine, D is not the answer. Because according to the second part of the question, it should be B, right? The first division segregation pattern happens. And finally, like if you look at these ratios, right? So these are what? 2:2:2:2:2. This is again 2:2:2:2:2. If you see here, this is 2:4:2. And this is also 2:4:2. So this is only happens when there is a crossing over, right? So to find out the distance, you know the formula. It is half into M2 SK, right? By total progeny into 100, right? So what is your total progeny? It is already mentioned. This is 300, right? So you have 300 here, but what will be half an M2 SK? So M2 segregation pattern happens or observed when there is a crossing over. So these will be M2 SK and half of them will be non-recombinant. So you will be multiplying it with half. And when you solve it, you will get 10 centimorgan. This further confirms your answer, right? So A here is centromere, B here is first division, and C here is 10 centimorgan. The explanation of the same I have mentioned here. Clear to you now, Ashu? It will be 10. No, it's fine, absolutely fine, Sunshine. It happens sometimes when we read the question, we are not able to properly interpret what is being asked in the question. So this is the reason we need to practice a lot of questions, right? Okay, that's great. Now you understood. That's great. Fine. So moving over to another question. So here answer is option B, right? Now moving over to another question. Now, in yeast, the His5 locus is a gene for an enzyme in the synthesis pathway for the amino acid histidine, and lysine level locus is a gene for an enzyme in the synthesis pathway for amino acid lysine. A haploid wild type strain, so genotype of the wild type strain is mentioned, is crossed with double mutant, and the diploid is allowed to undergo meiosis and 100 STR scored with the following results. So, what is the distance between the genes? Sahil, I hope you are able to follow the concepts now. So yeast, that means it is an example of what? Ordered or unordered? First of all, identify this. Okay, very quickly, some of you have solved this question. Suresh, Sarjan, Sunshine, option 2, you are saying. Very good. Let's check. Let's others also solve. Unordered, Sunshine. Very good. So unordered, what happens in unordered? Kumudi, yes, it is unordered. Very good. Okay, Deepshika, you're also saying option 2. Fine. Yes, even I discussed it in the class. You're absolutely right, Sunshine. Okay, Anu, 23. So basically, if you remember, when we talk about yeast, it's an example of unordered tetrad analysis. So basically, unordered means that here the arrangement of the ascospores in the ascus is not ordered. Like there's a lot of space in the ascus, because of that shuffling of the ascospores can happen. Now, for studying the unordered tetrad analysis, we have to consider here the two pair of alleles. So here you can see this is one parent which is wild type and another is double mutant. And when you are considering two pair of alleles, always remember three types of tetrads are possible. One is parental ditype, second is tetratype, and third is non-parental ditype. So parental ditype is like if you see here A plus B plus, right? So this is the genotype of this parent, right? A plus B plus, and this parent is having genotype AB, right? So obviously when these are crossed, you will get a double heterozygous condition, right? And finally, when when the diploid zygote undergo meiosis, three possibilities are there. Three types of tetrads are possible. So parental ditype is where you can see this is resembling to the parent one and this is resembling to parent two. So all four are parental, but because two are resembling to one parent and two are resembling to another, we say parental ditype. Now, if you see tetratype, here this is resembling to parent one, right? This one is resembling to parent two, but these two are recombinant, like A plus B and AB plus. None of the parent was A plus B or AB plus, right? So it is tetratype. All four are different, where two are parental type. Again, these two parents combinations are different. Genotype is different. And two are recombinant. Again, these two recombinants are having different genotype. Non-parental ditype is where you get all four haploid cells recombinant ones, right? But if you see the recombinant, these two are of one type and these two are of another type. So we call them as non-parental ditype. Non-parental because we are not getting any parental combination here, and ditype because non-parental. So why do we call them as non-parental? Because we are not getting any parental combinations here, and all are recombinants. And out of recombinants, two are of one type and two are of another. So non-parental ditype. So for solving this particular question, you need to recall the formula for calculating the recombination frequency in case of unordered tetrad, and that is half of number of tetra type ST plus number of NPD, that is non-parental ditype. Because what is the formula for recombination, calculation of recombination frequency? Number of recombinants by total progeny into 100. So if you see here, the recombinants you are getting in tetra type and in NPD. So this is the reason you have to write down the number of tetra type, and in tetra type also half are only recombinant. So this is the reason you will multiply here with half. Number of tetra type ST with half and NPD by total number into 100. So here the maximum, this 62. Always remember, the one trick here to remember is that most common are always parental types. Okay. And so this is parental ditype, and least commons are non-parental ditype. And these are then tetra. So now you can put the value, right? So it is half into 30, right? And then it is plus, you have 8 by total ST, which is how much it is? 100 multiply by 100. So half into 30, that is 15 plus 8 divided by 100 into 100, and that gives you 23 MU. Yes, it will give you 23 MU. So if you have answered option 2, you are absolutely right, which majority has answered. So really glad to know your answers. So you all are right. Very good. So to know such tricks, to know such concepts, as you all know, there is a Watson Junoon scholarship, right? So you can always, if you have already have one scholarship, you can always upgrade it to this Watson Junoon scholarship, because you get a lot of features in this particular scholarship. You get a 1 year coaching, beat any exam you're preparing for, either CSR NET, GATE, QET, IIT, JAM, or GAT-B. And even you get access to the 12 months of access to national workshops, global and national workshops, which we conduct. And also, like our programming, as you know, it's a very important internship. So here you, like a lot of platforms, they do this internship, but here with the Watson Junoon scholarship, in just minimum amount you are getting it along with this Watson Junoon scholarship, right? 4 certification courses of your choice you can select, any certification course. So four you can select, and that you can attend. And then also you will be getting direct access to our coaching experts on WhatsApp, and also a VIP pass to the CRISPR conference. And this is to know more details about Watson Junoon scholarship. You can apply, and you can contact, like you can, for more details, you can contact on these toll-free numbers, and you can apply anytime. So now moving over to the another question. Now this particular question is about the physical mapping. And I know, like I got a lot of questions from students also that, ma'am, please tell us the strategy about solving these type of questions. So believe me, first of all, these questions, they appear to be very vast, but they are very easy. If you know the trick, if you know the concept, you can easily answer such questions. So please go through the question and then you can tell me the answer. So here in the question, the location of 6 deletions, which are shown with these solid lines underneath the chromosome. Okay, these are the chromosome. They has been mapped to Drosophila chromosome as shown in the diagram. Now they are recessive mutations A, B, C, D, E, F, and G, which are known to be located in these regions of deletions. But we don't know the order of mutations on the chromosome. So when the flies homozygous for recessive mutations are crossed with flies homozygous for deletion, the following results were obtained, which you can see here, where M represents a mutant phenotype and plus represents the wild type. So you have to find out the relative order of the 7 mutant genes on chromosome. So take your time and answer this question. So quickly answer the question. See, more quickly you answer, more questions we can solve in this 1 hour session. Okay, so Kumar, Suresh, Sunshine, Rahul, you are answering option 3. Fine. So to answer this question, you need to be thorough with the concept of physical mapping. What is physical mapping? And this experiment, which is on which this question is based. Like here, the question is about when flies homozygous for recessive mutations crossed with flies homozygous for deletion. So what is this cross we are talking about? What is the deletion we are considering here, right? Okay, I think most of you have answered option 3. What about others? You just have to compare this particular diagram with the table. This is the trick here. Just compare the diagram with the given table. Now that you have to learn how to compare. So let me quickly tell you about the concept of physical mapping. Okay, so Ashu, you have answered option 2. Fine. Take your time. If you need some more time, you can take an answer. Okay, Murchana, option 3. Fine. So see, basically, I'm sure now you know about gene mapping because I've already talked about it in detail in the previous session. So we know that genetic map, when we talk about, they are based upon the recombination frequency, like linkage map. They are being plotted based on calculation of recombination frequency. But when we talk about physical maps, they are more efficient and more accurate because they provide you the actual position of the gene on the chromosome. And there are several techniques available for performing the physical mapping. So four main techniques are deletion mapping. We have somatic cell hybridization, we have in situ hybridization, and finally sequencing. So what is deletion mapping? So deletion mapping is a method where you determine the chromosomal location of a gene. And how you can do that? So for to make you understand that, I have taken this example, this cross example. So here, the individual which is homozygous for recessive mutation in a gene of interest, it is crossed with an individual that is heterozygous for deletion. So if you see here, this is the parent generation and this is the individual which is like with the mutation and it's in homozygous condition. And you can see that this another individual, it is having a deletion over here, right? This chromosome part is deleted where your allele A is present. So obviously, if these two individuals are crossed, what will happen? So if these two are fused together, basically if we are getting, so you can determine what will be the F1 generation. So basically, if these two are coming together, so we will get a wild type progeny. And here, if this is coming together and this is coming together, you will get a mutant progeny. Why mutant? Because here no wild type allele is there, right? So always remember, if in our crossing experiment, if you perform a cross and in F1 generation, you get one wild type and another mutant, that means your gene of interest is in the deletion region. So always in that case, half of the progeny will display the mutant phenotype. However, if you see over here, the alleles are present, right? This individual is having the mutant allele, this is having the wild type allele, right? So one is homozygous for the mutant allele and another is homozygous for wild type allele. And you can see here this chromosome region is deleted, but this is the region which does not contain your gene of interest. So if a cross happens between these two individuals, always remember you will get all wild type. So if the gene is not within the deletion region, all of the progeny will be wild type. That is what only you have to remember. Even I have mentioned the two key points here, which you can remember, that when recombination occur between two strains and you get a wild type gene, that means your mutation will is never in the region of deletion. However, if you get wild type, right, and mutant also, like mutant here you have to emphasize. So it means that your point mutation and the deletion are found within the same stretch of DNA. So in the question, it is mentioned that these are the deletions which are shown, right, in the chromosome. So what you need to do is, and these are the alleles A, B, C, D, E, F, G. We have to find out the order of the alleles. So here, this is, if I write it down, this chromosome here, so here the allele is absent. But if nothing is mentioned here, that means I can write plus. That means here the allele is present, right? Here it is absent. Okay. So because of the mutant means, mutant type we are getting means, anywhere you are getting mutant type, it means what? I told you that your point mutation and deletion are within the same stretch of DNA, right? So here you have a mutant. Then if you look here, this is mutant, but here we have like allele could be present, right? Then here you have mutant. So like this you have to, you can write down, right? So accordingly, you have to just compare. Like if you remember, I told you the trick. You have to compare this table with that of this diagram which is given. So what do you think is the answer here? So here this is mutant, here it is present. So what is your answer? Some are saying option 3, some are saying option 2. Okay. All have answered. So let's check. So as I told you, by overlapping the deletions and comparing the loss of function genes, we can predict the sequence. So it should be B, C, D, E, A, F, G. So here you can see M plus 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 plus. So this is the pattern shown here. So this must be B. Then M, M, and then 4 times plus it is there. So that means this should be C. Then we have, like if you see here, this is M, this is M, this is M, this is M, then plus and then M. So that means this is the pattern shown here in D, right? So accordingly, you can answer like here plus, like for here it is plus, here it is for deletion 2. So no deletion plus, but here M, M, and here you will have the plus and then M. So this is resembling to E, right? This one. And then if you look here, this will be plus, 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 plus. Then you have, so you have like this way. Just a minute. So you can write it down for all the combinations where you are seeing this pattern, where you are seeing this pattern, only that you have to compare. Okay. So like plus, 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 M, 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 right? So plus, plus, plus here, M, 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 right? And then here you can see plus, 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 M. So that means this is A and this is M here. So this is F and this is last one is your G, right? Because this is plus, 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 and then here you have M and here you have plus. Clear to everyone? So my answer here is option 3. And Ashu, is it clear to you? Then moving on to another question, that following scheme represents deletions 1 to 4 in the R2 locus of phase T4 from a common reference point. So again, 4 deletions are here and 4-point mutations A to D is there, which are tested again 4 deletions for the ability or inability to give the wild type recombinants. And the results are summarized in the table, and based on the above predicted order of the point mutation, you have to find out. Is it option 1? Is it option 2? Is it 3? Or is it 4? So find out again. You have to just match the this deletion profile with that of the table which is given. So there are some deletions 1 to 4 which is shown here in the R2 locus of page T4, and 4-point mutation A to D are tested against 4 deletion to for their ability to or inability to give wild type. So you know wild type you can only get when your the mutant allele is not in the deletion region, right? If you are getting mutant, that means the pointer that is your mutant and the region of deletion lies in the same region. So what is the answer here? So from here it is like all inability, right? Minus, right? So if here something is not there, that means we can expect like here you can see. So that means here it can be plus. So we can write the pattern plus minus minus. This way you have to do. Okay, I'm just helping you out. Think about it. So plus minus minus minus. What could be the another pattern? Plus plus minus minus. Third pattern plus plus. What could be the another pattern? Draw it. Try and then tell me. So this is plus plus minus minus. Third could be plus 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 minus. Yes or no? And then finally you can have plus 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 plus. Yes, it is option 2. So this could be D, because if you see D is having this pattern. Then if you see here, this pattern is given by B, plus plus minus minus, right? Then if you see the third one, plus 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 minus, it should be A. And if you see the last one, it should be resembling to C. So DBAC is the answer. Clear to all now? Yes, very good. Everyone has answered it correctly. Very good. Now moving on to another question. Now this question is based on somatic cell hybridization. A series of cell lines was created by fusing mouse and human somatic cells. In mouse human somatic cell hybrids, human chromosome tend to get lost before becoming a stable cell line. Some hybrid cell lines may carry human chromosome deletions, and each cell line was examined for the presence of chromosome and for the production of an enzyme. The following results were obtained. So you have to answer which segment of the chromosome has the gene encoding for the enzyme. Now to answer this question, you should have a like thorough knowledge of somatic cell hybridization. What happens in somatic cell hybridization? So although again these questions appear to be very difficult, but they are very easy. Believe me. So what do you think is the answer here? Okay, option 2, option 3, again getting mixed answers. Okay, majority says option 2. Take your time. Think about it. What is the answer here? What is the trick here to solve the question? Okay, so majority says it is option 2. Okay, Rahul, you are saying option 3. Fine. Done, everyone? Have answered? Fine. So basically, let me just tell you in brief about somatic cell hybridization. So basically, when you fuse two different types of cells, okay, like human fibroblast and mouse tumor cells, when you mix, so like these are cell lines, like these are the like from a single cells, you are growing the cells in a media and they are maintained for a particular time period. So like what are cell lines? These are the population of the cells which are maintained for a particular time, right? In a suitable media. So here you can see human fibroblast and the mouse tumor cells, they are mixed. And obviously for the fusion to happen, you need to provide some sort of trigger, some sort of chemical reagent. So polyethylene glycol is provided and that results in fusion of these two cells. So you can see these two cells, when they fuse, they result into formation of a structure which is called as heterokaryon. But you can see in this heterokaryon, the nucleus of both the cells are still not fused. So later on, what happens once the heterokaryon is formed, human and mouse nuclei, they can fuse together and resulting into the hybrid cell. So this is the hybrid cell with the fused nuclei. Now it is observed that during when these hybrid cells are formed, one of the species chromosome is lost, and mainly it is human chromosome. So human chromosomes are randomly lost from the nucleus. So not all the chromosomes, some chromosomes are lost. Now maybe in different cell lines, different chromosomes can be uh, lost. So human chromosomes are randomly lost from the nucleus during cell division. And finally, like here you can see in the figure, we have A to E cell line. So maybe in A cell line, we have chromosome number 2 and 4, which is lost. In B, maybe it's chromosome 1 and 6, which is lost, right? And so on. So different human chromosomes are lost in different cell lines. So if you have to identify whether your gene of interest, that is a chromosome carrying your gene of interest, is present or not, how you can detect? So either we can look for the product which is encoded by that gene. Let's say a gene encode for a particular enzyme. If you are getting that enzyme in the like if you are seeing that enzyme, that means your gene is present, and gene presence means that obviously that chromosome is present over here, right? So basically, the which gene is present on which chromosome, we can easily find out. All you need to do is you have to first find out that, like either you can look for the protein which is produced by the gene, or you can even directly look for the gene itself by using the probes. Like you use a oligonucleotide complementary to the sequence of your gene of interest, and you can label that probe and you can visualize the presence of your gene of interest, right? So if we can correlate the presence of gene with the presence of specific chromosome, we can allow the gene to assign to a correct chromosome, right? Like for example, let's say you have cell line A and you have cell line B. Cell line A has chromosome number 2, 4 and 11, and you have in cell line B, you have chromosome number 2 and 11. Now let's say you are looking for the product of gene A. So if you are able to see the product, like let's say the cell line A shows the product, but cell line B doesn't show the product. So what does it indicate? What is the difference between these two cell lines? They differ in chromosome number 4. So this indicates, since we are getting the product of gene here, it means that gene is present, and present on which chromosome? It's chromosome 4. So like this, you can find out the correlation between the presence of gene, its product, by looking at its product, or and with the presence of specific human chromosome. So over here in this question, we have it is like all you need to do is you have to just see over here that the gene product is mentioned and cell line is mentioned. So gene product you can see is present in A cell line, B cell line, and D cell line, but absent in C and E. So that means whichever chromosome is carrying the, like having the gene, will be also showing the product only, right? It will exactly match with that of this pattern, right? So if you see 1p, right? So it is showing the presence of the gene product in A cell line, C and E, not in B and D, right? So it doesn't match. So like this, you have to match where you are seeing this pattern, plus, plus, and minus, plus, minus. Only this thing you have to check. So if you look here, I think here we can see, like we have the option 1p. So 1p is not showing. If you check 5p, so yes, 5p is exactly showing this pattern. Plus plus, that is gene product presence for A and B cell line, not for C, not for E, but for D. And if you see 5q, it is showing plus 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 minus minus. So again, this cannot be the answer. And 4p, if you see, again, this cannot be the answer. So you can see it's a very easy question. We can directly say that that means the answer is here option 2, because definitely if we are getting a gene product, it indicates the gene is present. And all the one thing that we have to remember here is that the although you can see presence of gene product is in other chromosomes are also showing it, but it is not exactly matching to this profile of this cell line profile, that you have to keep it. Now, moving over to another question. So, Rahul, I hope it is clear why the answer here is option 2 and others have answered it correctly. So, this is the last question we are going to discuss today. So, quickly go through this question and tell me the answer. This is again based on somatic cell hybridization. Three somatic hybrid cell lines designated as X, Y, and Z have been scored for the presence or absence of chromosome 1 through 8. Okay, so you can see this is chromosome 1 to 8, which is mentioned here, as well as for the ability to produce the hypothetical gene products. A, B, C, and D are the gene products. And these are the cell line X, Y, and Z. Which of the following option appropriately assign chromosomes for each of the given genes? So first of all, just to look at the table, just relate, just keep in mind what I told you, right? The matching you have to do. The gene expressed means its product should be there, and definitely the chromosomes which are there, so you can correlate them. If the gene is present, that means uh, like you can assign it to the chromosome in which it is present by looking at the gene product. So what do you think is the answer here? Please quickly answer it. What do you think? Gene A is present on which chromosome? We have total 4 genes, right? Gene A, gene B, gene C, and gene D. So gene A is present on which chromosome? It is minus, like gene product is absent in X cell line, but Y and Z it is present. So here in boxes where if nothing is mentioned, that means it is minus. Okay, this is also minus, 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 and here also it is minus. So minus, plus, plus for gene A, right? For B, it should be plus, minus, plus. For C, it should be minus, 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 and for D, it should be plus, plus, plus. Okay, Aishwarya, you are confused between option 1 and 4. Fine. And others, you are saying option 1 it is, right? Fine. So basically, you all can see here that minus plus plus. So where we are getting this pattern, this is here, right? So definitely we can say here that your gene A is located on chromosome 5, right? So gene A must be on chromosome 5. Then if you see B pattern here, so plus minus plus. So it is shown over here, plus minus plus. So B we can say it is present on chromosome number 3. Then C will take later. Let's talk about D. So D it is plus plus plus. So definitely it is on chromosome number 1, right? And so where these options are given, so if you see, answer cannot be option 2 and option 3, right? Because according to this, gene A is on chromosome 4, and according to this, the gene A is on chromosome 5 and B, but it is not talking about D, right? Which is even mentioned here. Now, if you compare option 1 and 4, they only differ in gene C. So here it is mentioned gene C is present on 8. Please remember, absence of the gene will not confirm anything. Presence of gene product confirms anything. So although here up to 8 chromosome is mentioned and you can say that, okay, C is present on 8, So this is incorrect because maybe the gene C product, maybe this gene C could be present in some other chromosome. There are many chromosomes in humans, right? We have 23 we inherit from one parent, 23 from another, 22 autosomes are there, right? So definitely we cannot conclude that C is on 8. It is not showing any presence. It is the absence only, right? So definitely this could not be the answer. 4 is correct here and not 1. Okay. So answer here is option 4. 1 is not correct. I hope it is clear to you, Aishwarya, now. So with this, we have come to the end of the session. Do you really like this session? Do you think it was really helpful to you? Yes. Do you think now you can solve the questions based on these topics? I hope you have liked the session. Yes. Did you like the session? Okay, that's great. Thank you so much for your responses. So in the end, I just want to say, just practice as many questions as you can. And even if in the beginning, maybe you're not able to answer, you're not getting all the answers, you're not able to get the correct answers for the questions. So don't lose confidence, just practice. Just remember, victory is always possible for the person who refuses to stop fighting. So this is all for now. Thank you, everyone. We'll see you in another session.